Are you on mute? No. Großartig. Thank you, Logan.
Hey, it's me. Call me when you can. Let me paint this picture for you, baby. It's been nice so long, and he never comes home. And every time you call him, all you get's a busy tone. I heard you found out that he's doing to you what you did to me. Ain't that the way it goes? When you cheated, girl, my heart beat, girl. So we go without saying that you left me feeling hurt. Just a classic case scenario. Till there's always time, girl, you got what you deserve. And now you want somebody to kill them all the nights. Wish you had somebody to right, girl, I ain't somebody really out of sympathy. Don't you take see, should listen to me, baby.
Welcome to Metabolic Physiology in Isolation, MetPhys 2020. I've said this every day, but I'm going to reiterate it today. I think there's been some talk on Twitter, and we're going to make sure people understand the value of the model. It's not just that we have an online seminar series that some can attend. It's that it's free to attend, it's inclusive because it's accessible. We make it accessible by recording every talk and placing them online at 2 separate sites so people, wherever they are, can access them for free at any time that they're able to listen. In these days of the COVID-19 crisis, a lot of us have many other responsibilities. Many of you work in hospitals, you have your children at home, and the time that the conference airs is not the time that you can watch it. By making it available online and forever, we actually make this silence truly inclusive. Anyone can watch it anywhere, anytime, as long as they have a computer and an internet connection, or a phone and an internet connection, because it's on YouTube, Facebook. These are platforms most of us have access to. The great thing is it's also green. We're not traveling anywhere. We're not bringing anyone anywhere. I do not think this model will replace in-person meetings. There is so much benefit to actually meeting our colleagues in person. However, I really like the fact that a lot of people who couldn't get to those conferences because of other reasons, they have family responsibilities, they can't meet the cost, they're early in their graduate career and haven't got data to share, can listen to cutting edge science at any time they please. And they can present their work too. There are people that are presenting who may have responsibilities where they wouldn't be able to get away from home to come and talk. And we're able to feature them because of this model. So I really do think this is the future of conferences. I love the fact that anyone can access it, and I'm glad that you all are on board. If you're listening to other science talks and for some reason they have a reason not to make the recordings public, at them, request this. I think this is not too much to ask. I don't think there's a reason any scientist should be giving a talk that's public, but they're not sharing it with anyone who's able to access it. There's no reason for exclusion in science, and I think inclusion is the way to move forward. I think that's going to remove one of the important barriers to diversity in science, and I think this is truly important. So if you have other series you're trying to follow, but you can't access them, contact the organization. Make sure they know that it's important to you, that it's inclusive and accessible. Accessibility is key to that. That's what we're trying to maintain here for you. Nearly every day I've gone through and found some fantastic stuff on Twitter. It's not that there wasn't yesterday, but you know what? I'm just so impressed by Cassidy and her ability to take a photo in front of a photo in front of a photo. And I think we really can't appreciate that unless we make that photo big. But yes, she's gotten 11 days in a row, and I bet right now she's taking another photo with 12 in a row. If anyone has ever drunk tea before, you know that is perfect little finger teacup holding. I think we can all aspire to have that kind of technique when we drink tea. I know I sure am. So good on you, Cassidy. We're all impressed by your ability to stick with it. We're going to see you all the way out to April 24, I have a feeling, and I'm excited to see that. None of this would be possible without our truly amazing lineup of speakers who not only have agreed to present their unpublished work, but also have agreed for it to be openly accessible on multiple platforms for everybody. And I really think that, you know, a part of the reason this works is because our speakers have gotten on board with the accessibility and the inclusion. And I really want to thank them for that. I think you can thank them in person. Remember these faces, not just these faces, but I released the new lineup today. I released the full schedule with the talk titles, and I think you are all getting excited about how wonderful MetPhys 2020 is going to be. We are finishing on April 24, but I am going to launch MetPhys 2021 next year, and we already have a pretty good speaker lineup already in the books. So stay tuned. This is not a once-off. This is something that's going to move forward, and I think this is going to be great for everybody who enjoys cutting-edge science. So here are our new list of speakers that are taking us all the way out to April 24. They are fantastic scientists with really great stories to tell and covering a diversity of metabolism field. And I think you all are going to enjoy those. If you haven't heard of them before, make sure you tune in and listen. Don't just tune into the ones you know, because I assure you that all these people were handpicked because they're doing cutting edge science right now. If you'd like to thank me personally, I wouldn't be in the position I am without the support from the Pathway to Stop Diabetes program. One of our speakers later in the program, Ciliary, is also supported by this program, and I think you're going to be convinced after you hear her talk of how important this program is. If you'd like to help us or support the Pathway program, a small donation, $1 or $5 in our name, would be a tremendous honor to us. You don't need to do that. You can just enjoy the talks. And if you really want to share your enjoyment of the talks, hit us up in the Twitter space, use the hashtag MetPhys2020 and ask questions or make comments. We're all enjoying that. And I know a lot of the community is just enjoying the community atmosphere, the sharing of photos, the sharing of cats, dogs, very good best boys, coffees. And just seeing the stats on Felix Vogel's cycling was pretty impressive to me yesterday. I asked for graphs and he supplied them. I think he's going to be the best in shape person at the end of this. Unfortunately, I'm going the other way. I'm fattening my curve, but that's all right. So today we have Jantje Gerdes. telling us about cilia, but tomorrow we have another amazing talk. Isaac Harris is going to step up to the plate and talk about the roles of antioxidants in cancer. And that stands to be a fantastic talk. So anyway, I've talked enough this morning. I am going to pass the reins over to Jantje and in Munich, in Germany. And I'm sure you're going to really enjoy this talk. So let me just make her the host. All right, take it away, Jantje.
All right. I hope everybody can hear me now. I'm just waiting for Phil to DM me to see if everything is working properly. So in the meantime, let me just tell you a little bit about what we're doing. For all those of you who are not as familiar with primary cilia, I'm showing a couple of examples of the cilia that we are particularly interested in. On the top right-hand side, the panel is a murine islet, isolated pancreatic islets with insulin stained in red and primary cilia stained in green. We can find primary cilia in mouse islets on beta cells, alpha cells, and delta cells. And we can also see it in the electron micrograph. This was supplied to us by Andreas Müller very kindly from the Paul Langerhans Institute in Dresden. He's also on Twitter, so if you want to see more beautiful images of beta cells and pancreatic islets, take it away. I'm on Twitter. My Twitter handle is shown here. So if you have questions, hit me up. I will hop on Twitter after this talk and see if I can answer your questions. Sorry. So I want to start this talk with acknowledging the people who did most of the work. This is my lab, and primarily the stories that I'm presenting are done by Francesco, who's the first author of a recent Nature Communications publication that we had out in December. Francesco Volta did his PhD in my lab, and he's now moved on to do a postdoc in his native Parma in northern Italy. So as I'm sure everybody is aware, this is one of the hardest hit regions with the COVID-19 disease, and our thoughts are with him. So far, he's doing well, and his family is healthy as well. And we certainly hope that this is going to continue that way. Julia is a postdoc in my lab, and she had a baby last year. She was going to return from a parental leave. Baby Theo was in daycare for exactly 1 week before it had to close down due to COVID-19. So that has been a little bit of a bummer, but you know, as long as we are all healthy, it's fine. And then Annette, who is a stellar technician in my lab and really has contributed to most of the stories coming out of our lab significantly. She just had a baby on March 22nd. Baby Fred was born in isolation, and his daddy couldn't be with them afterwards while in the hospital, but I'm happy to report that the whole family is at home and safe now. So in varying degrees, we're all affected by the coronavirus, and so are actually cilia. This is an image that I lifted from the INSERM homepage in Paris today, and you can see here cilia, ciliary axonemes with the microtubule doublets and pseudo-colored coronaviruses here. So there's a close proximity between these motile cilia and the viruses, whatever that may mean, but there was an early report coming out of the Wuhan cohort that motile cilia beat slowly, or there's no beating of these cilia anymore in patients with COVID-19. And because they're important to move around mucus and other components in the lung and in the tracheae, that could mean that this is significantly contributing to the pneumonia that's associated with COVID-19. But I promised that was the last slide about coronavirus, not only because I want to take our mind off this disease for a while, but also because I'm not an epidemiologist or a virologist, so I cannot speak with any authority on this disease. I want to show you a couple of other examples of cilia. Top left is the paramecium, and this is the only non-mammalian example that we show, or that I'm showing, of cilia. Here are mammalian tracheae. So these are the motile cilia that I was talking about before. And the other examples are sensory or primary cilia. There's only one cilium per cell, as opposed to the motile cilia, where there can be more than hundreds cilia per cell. And they appear in a number of different tissues, such as the kidney epithelium and the pancreatic epithelium, but also in neurons, odontoblasts, and other tissues. And this is an important point that I want to make, because cilia in invertebrates are mostly found in sensory neurons and in the reproductive organs. Sperm, for example, have a flagellum that closely resembles a motile cilium in the structure. But in vertebrates or mammals, we actually see many more tissues where we can find primary cilia. And you name it, you'll find it in the pancreas. I've just shown you. We've seen it in the epithelium, but also, as I've shown on the top slide, in islet cells. We see cilia also in the liver, but interestingly, they're not in hepatocytes, they're in cholangiocytes. And also in other cells such as heart myocardium, and also in skeletal muscle, we find some cilia. The structure of the cilium is fairly well conserved. It is a circular arrangement of 9 microtubule doublets. And in sensory cilia, there is no central pair of microtubules, so these are the 9+0 cilia, whereas in the motile cilia that I've shown you in the previous slides have a central pair of microtubules, and they are actually motile, whereas the primary cilia are not thought to be motile. Another important point that has emerged over the last decade or so is that the cilium is really a signaling hub. There's many different pathways that have been associated with the cilia and ciliary function, and one of the gold standards still is today the hedgehog signaling pathway. But there's other pathways as well. For example, PDGFR alpha-alpha. It has been shown that the homodimer of PDGFR alpha has to be localized to the cilium for the cell to be able to re-enter the cell cycle. But also beta-arrestin has been found on the cilium. This is a publication by the Lefkowitz lab. And we've heard yesterday about the importance of several GPCRs in metabolism. So this might also be important for other GPCRs. Other receptors that are localized to the cilium are serotonin receptor, somatostatin receptor, SSTR3, and melanin concentrating hormone receptor, MCHR1. The disease that I was interested in since my postdoc, which I did with Nico Katsanis, who is now at Duke, but at that point was at Johns Hopkins University, was Bardet-Biedl syndrome. Bardet-Biedl syndrome is a fairly rare disease. It occurs in roughly 100 to 170,000 live births, but it can be more frequent in certain populations, such as in Newfoundland, for example. And the core symptoms are retinal dystrophy. So this can be fairly mild, for example, by night vision, but it can also lead to blindness. And post-axial polydactyly, interestingly, it's always post-axial, so it's an extra pinky, but not an extra thumb. Other major features include obesity. It's also called often or referred to as the obesity syndrome, and varying malformations. But also the minor features, which are not core symptoms of the of BBS or Bardet-Biedl syndromes, are schizophrenia and diabetes, which is why I was particularly intrigued by this disease. Bardet-Biedl syndrome is one of several ciliopathies. This is a class of diseases that is caused by mutations in ciliary genes, and there's a number of different genes that are involved in ciliary function. The BBS genes that I'm mostly interested in are localized to a multi-protein complex at the base of the cilium. Today we know of 21 different genes that, if mutated, can give rise to Bardet-Biedl syndrome. And the organ involvement varies among the ciliopathy genes. Very common are cystic kidneys, retinal degeneration that I've mentioned before, anosmia, liver fibrosis, mental retardation, and infertility due to a motile sperm. And the class of diseases that I was particularly interested in since my postdoc was obesity and diabetes. And one of the reasons why we were really studying Bardet-Biedl syndrome is because it's an oligogenic disease that I mentioned before. It's a Mendelian, non-Mendelian path of inheritance. Some of that is Mendelian as well, but it's complex. 21 different genes can give rise to it. Some evidence of triallelism is there. And so we were thinking maybe we can learn by studying Bardet-Biedl syndrome something about the more complex traits such as obesity or diabetes type 2. And Philippe Frogel's lab actually showed that variants in BBS2, 4, and 6 are associated with obesity, either childhood or adult onset common obesity in a French Caucasian population, showing that this might actually be the case. So then the question was for me, can oligogenic ciliopathies inform us about type 2 diabetes? And before I really launch into diabetes, I want to show just two examples of more recent research into the link between primary cilia and obesity. One of them showing that it's really the hypothalamus that seems to be at play here in the satiety signaling, as well as in the opposite in hunger signaling. So the BB-some in POMC and AGRP neurons is necessary to regulate body weight. And the metabolic receptors that are involved apparently in sorting or of the BB-some to or from the cilium are, among others, neuropeptide Y receptor, NPY2R, and serotonin receptor. Another study that was published late in 2018, I thought was really interesting because or 17, sorry, it showed that very commonly associated genes that are linked to obesity, like the melanocortin concentrating melanocortin 4 receptor, as well as the adenylyl cyclase 3, are mutated in areas that are important for ciliary localization. So cilia might actually be underlying common pathways for genetic predisposition to obesity and not be as rare as some might think. And a final point about neurons I want to make is a recent study that was published in 2019 by Rafael Rodrigo and co-workers. And what they showed by metabolic labeling is that cilia in L2 neurons, for example, are very old. They can be as old as the organism that they're found in, which is probably reflected of the very low turnover of these neurons. But what I found even more interesting, because I care mostly about beta cells, is that the cilia in beta cells are also very old structures or very enriched in these labels, which is again probably reflective of the low turnover. And what this means is that perhaps cilia are more vulnerable in these cells to metabolic insults or any other kind of injuries compared to other more regenerative diseases or regenerative tissues, because this is because this might be low turnover and very hard to repair for the cell. So we have done, or I have done some work before I started my own lab in Per-Olof Berggren's lab in Stockholm. And what we looked at there was at glucose handling in BBS4 knockout mice. We've found that as early as 7 to 9 weeks of age, when the mice are still weighing the same, we see impaired glucose handling in BBS4 knockout mice already. And this is linked to almost complete blunting of first phase insulin secretion in these animals, suggesting that really there is a role for BBS4 and associated or other ciliary proteins in insulin secretion. What we've also found was that there are ciliary defects in a type 2 diabetic model. In this case, it was Koto-Kakizaki rats, and what you can see here are islets, and in green, the cilia of these islets. And what we found is that there's significantly less ciliated beta cells in GK rat islets compared to Wistar islets, which is the strain that they're derived from. So when I started my lab, I was going to look more closely into the role for primary cilia in islet cells, such as beta cells, and thus insulin secretion. And I want to spend a little bit of time on the role of these islets of cilia in beta cell function. And what Francesco, as I said, he's the head of the study, has done was he generated beta cell specific inducible ciliary knockout mice. And because this is a mouthful to say every single time, we call them BICKO mice for short. So he did this by placing floxed allele of IFT88, a core ciliary component, under the CRE-ER that is under transcriptional control of PDX1. We induced the recombination at P25 to 35, so roughly 4 weeks after birth, and then followed the phenotype over a total of 20 weeks. And what we found was that as early as 4 weeks after induction, we see glucose intolerance, and then this gets progressively worse. So 12 weeks after post-induction, we see really very dramatic glucose intolerance in these animals compared to the control mice. This is true both for the blood glucose excursion as well as the area under the curve. And we see also that this is due to a complete blunting of glucose-stimulated insulin secretion. Interestingly, though, this is happening at a time, so as I said, 4 weeks after induction, we already see glucose intolerance, but 6 weeks post-induction, so 2 weeks after the first measurements, we don't see any evidence of loss of beta cells in these mice. The picture is completely different 14 weeks after that, so 20 weeks post-induction. We see more than 6-fold reduction of beta cell mass in these mice, and this might be due to a increased level of apoptosis in the beta cells in these islets in tamoxifen-treated BICKO mice. What we have shown previously was that primary cilia are involved in insulin receptor dependent signaling and insulin secretion in beta cells. But when we looked at the phenotype that I've just shown you, we thought that insulin receptor itself or alone can probably not explain the phenotype, because the beta cell specific insulin receptor knockout mice, BICKO mice, are not as severely affected by this deletion of insulin signaling from the beta cells. Only 25% roughly of the mice develop diabetes or glucose intolerance. So what Francesco did was he ran a receptor tyrosine kinase antibody array, and what he found was that there is a significant upregulation of phosphorylation of the FA2 and FA3 receptors. And consistent with our finding, we also found the ERK-MAP kinase pathway upregulated, which is a downstream target of FA2, FA3 signaling. We cross-referenced the BICKO data with an antibody array from BBS4 knockout mice. And we also found significant upregulation of FA3 and FA2 phosphorylation, as well as hyperactivation of the downstream ERK-MAP kinase pathway. It has been shown in 2007 by Eki Lamat's lab that FA-efferent signaling is regulating insulin secretions, so we thought that this was a good candidate to pursue further. And Francesco confirmed that there was hyperphosphorylation of FA3 in BICKO islets that were treated with tamoxifen compared to the vehicle control. He also generated stable cell lines that were depleted of IFT88, so these are 2 clonal cell lines. Depleted of IFT88, and they also show hyperphosphorylation compared to the controls. Consistent with what we've shown before, we also find hyperphosphorylation of the ERK-MAP kinase downstream target, both in the primary islets as well as in the phospho-ERK pathway in the Min6 cells. So we thought this was a good model to study further, and FA3 was a good candidate. But first, to show that really it is FA-efferent signaling that's at the base of the phenotype that we observed, we treated islets isolated from tamoxifen-treated BICKO mice compared to controls with recombinant efferent A5 protein. And shown here, you can see in gray the level of the tamoxifen-treated mice that were not treated with efferent A5, and shown in black, you see the wild-type excursion, and you see that there's a severe blunting of glucose-stimulated insulin secretion. But when we treat these two different islets with recombinant efferent A5, we can restore this insulin secretion phenotype. And we can see this again, both in the excursion here of the dynamic glucose-stimulated insulin release, as well as in the first phase and total secretion when we add the secretion up. We further manipulated the FA-efferent signaling pathway to really convince us that this was at the base of the phenotype that we observed. But first we had to show that this was specific to IFT88. So we turned to the Min6 cells and just looked for glucose-stimulated insulin secretion in these cells. And you can see here that overexpression of IFT88 in the IFT88 depleted cells restores the stimulation index back to those of control Min6 cells. And also treatment with efferent A5 restores the stimulation index, suggesting that this is really at the base of the phenotype. We also received some dominant negative FA5 construct from Eki, who was really helpful in trying to figure out the phenotype. And what we found here was that not only can we restore the insulin secretion in response to a glucose stimulus in these, but we also deregulate basal insulin release in the absence of high glucose, so in low glucose, suggesting that really FA5 or efferent signaling is regulating insulin release. But it's actually fairly uncommon that we see upregulation of a signaling pathway compared to downregulation. So there's of course several ways of achieving increased signal. One of them would be to have higher input, and one of them would be to reduce the negative feedback regulation that we see. And it has been shown that receptor tyrosine kinases do spontaneously autophosphorylate even in absence of ligand. And to suppress this autophosphorylation, they are internalized and transported to phosphatase to dephosphorylate them and basically suppress the spontaneous signal coming from them. And in the case of FA-efferent signaling, this is a phosphatase called PTP1B, which resides in the perinuclear region. So to test our hypothesis, what we did, or what Julia did, this is actually Julia's experiment, was we did a pulse chase biotinylation, surface biotinylation assay. And what Julia found in the first step, when we she just biotinylated the surface proteins of our cells, the Min6 cells depleted of IFT88 or controlled cells, was that there was a significant upregulation of phosphorylation levels of FA3, but not of global FA3 at the plasma membrane surface. So the next step was to let the cells go further for an hour so they can actually internalize ligand that's present on the surface and deplete them of biotin, strip them of biotin. So at this point, we will only see internalized protein labeled with biotin. And what she found was that in this case, there's less phosphorylated FA3 compared to the control cells, suggesting that there's really a lack of internalization, or a lag of internalization at least. When she let the cells go for an additional hour, stripped them again of biotin. So at this point, we will not see protein that has been returned to the surface, so recycled protein, and only the internalized protein will still have a biotin label. We see that there's increased levels not only of FA3 in the IFT88 depleted cells, but we also see increased levels of phosphorylation of FA3, suggesting that this is not only a defect of internalization, but this defect must occur before the ligands reach the the perinuclear region where the phosphatase PTP1B resides. Another way of showing this was when Francesco checked in FA3 internalization. So what he did, he supplied the cells with labeled Ephrin A5 recombinant protein and measured the internalization by measuring the total internal fluorescence. And what he found was that with a delay of 15 minutes, control cells efficiently internalize Ephrin A5, which is a ubiquitous ligand and binds to all FA receptors. But by comparison, the internalization is much slower in the IFT88 depleted cells, and also less compartments are occupied by the labeled ligand. He also showed this in primary islets from the BICKO mice, suggesting that this is really happening not only in the cell line, but also in the primary islets. So I've mentioned PTP1B before, which is the phosphatase that is involved in dephosphorylating FA3 or FAs in that case. And you can see here by overexpression of PN1, which is the gene that's encoding for PTP1B, it's flag tagged, and overexpression shows a nice perinuclear localization, but probably because of the overexpression, we can also see it in the membrane compartment, or close to the membrane compartment of these cells. When Francesco treated the cells with different inhibitors, one of them was a specific inhibitor for PTP1B, and one of them was a pan-PTP inhibitor that is inhibiting all protein phosphatases. We interestingly did not observe any difference anymore in phosphorylation, suggesting that really at the base of the defect that we observed is a defect in dephosphorylation, not increased phosphorylation. But overall, the levels of phosphorylation are reduced when we use a PTP1B inhibitor compared to a pan-PTP inhibitor, suggesting that maybe there's another phosphatase involved in the communication or in the regulation of ephrin signals. And I mentioned PTP1B before, so this is a phrase that I liked from Francesco. He said, if FA3 cannot reach PTP1B, we have to bring PTP1B to FA3. So this is actually the overexpression experiment that I mentioned before. And we took advantage of the fact that we do see levels of PTP1B at the plasma membrane. And indeed, we can rescue the insulin secretion phenotype with overexpression of PTPN1, suggesting that this is really at the base of the phenotype. So I find this really exciting because it suggests not only that there's different phosphatases involved, but also that the receptor is processed differently in the endosomes depending on whether it's bound to ligand or not. Not only were we surprised to find FA3 because of the hyperphosphorylation, but ephrin is actually a juxtacrine signaling pathway. So it was a little puzzling to us that we saw this in conjunction with the cilium, but the internalization defect might explain why we see this. And on top of that, what we see when we treat the cells with ephrin A5, so this is a ligand of FA3, but also other FA receptors, we see that there's significantly decreased levels of hyperphosphorylation in the IFT88 depleted cells compared to the controls that have not been treated with ephrin A5, suggesting really that the ligand-bound receptor is processed differently and can recruit an additional phosphatase as opposed to the just PTP1B in the control case. This is also true for primary islets, suggesting that this is not only happening in the insulinoma cell line, but this is also relevant for other cells. So moving on, we now know that probably the defects that we see are due to endosomal processing or interference with endosomal processing on one specific phenotype or pathway, which is the internalization for dephosphorylation in absence of a ligand, so it's just autophosphorylation. But how can we explain this? And for this, we overexpressed a MYC-tagged form of FA3 because we couldn't find an antibody that was suitable for FA3 staining and visualization. And what Francesco found that in the control cells overexpressing MYC-tagged FA3, we see a cytosolic distribution, clear distribution of FA3, much like one would expect. And when we treat with FNA5, this becomes even more prominent, localization in the perinuclear region and the cytosol. However, when we stimulate IFT88 depleted cells, it seems like the MYC-tagged FA3 gets stuck in the periphery, close to the membrane of the cells. And this is the area also where F-actin, for example, would localize in the periphery. And because it's known that cilia and actin dynamics are closely related, we were thinking about the F to G actin ratio. So filamentous actin is polymerized and G actin is the monomeric form of actin. And what we see is there is a shift towards more filamentous actin in the F to G actin ratio in IFT88 depleted cells. And this is also echoed in the immunostaining with phalloidin and in IFT88 depleted cells. We see here more and thicker stress fibers that are, however, shorter compared to the knockout, to the controls. And this has been reported in other examples for BBS proteins as well. So it seems to be really common to cilia, not just to IFT88. And because this is reminiscent of delamination, which in turn could be indicative of epithelium-mesenchymal transition, we were looking into EMT markers. And first, I found during my postdoc in Nico's lab that when we deplete ciliary proteins, such as BBS4 in that case, we actually stabilize beta-catenin and thus have an increase in beta-catenin-dependent canonical Wnt signaling. And what we saw here is that indeed also when we deplete IFT88 from beta cells or insulinoma cells, we see stabilized beta-catenin, which in turn could induce an EMT transition. So we looked into mesenchymal markers, snail-1, slug, and vimentin, and we found an increase in all three of those, and maybe even more important, we see a drastic reduction in E-cadherin, which is an epithelial marker, suggesting really that there is a polarity phenotype linked to this. And the epithelium mesenchymal transition might cause the defects in the actin reorganization. So how is this linked? One of the first important steps of actin reorganization and then internalization of ligands is RAC-mediated actin reorganization. So we tested, or Francesco tested, the GTP-bound levels of RAC1 in IFT88 depleted Min6 cells. And what he found was that there is an increase of GTP, or active RAC1, in the IFT88 depleted cells. And that addition of FNA5, which induces RAC1 activity and then rapid hydrolysis of GTP to GDP, does not make a difference. So there is no decrease in GTP-bound RAC1 or no activity. When we looked into what might be the cause of this, we looked into the GEF of RAC1, which is TM1, among other things. And what we found was there is a drastic upregulation, in this case shown here, mRNA, but also protein, both in the Min6 M9 cells as well as some BICKO islets. And the potential link between TM1 and polarity might be stemming from the fact that TM1 is complexing, is a polarity complex consisting of PAR3, PAR6, and atypical PKC, and then is mediating the exchange of GTP to GDP of RAC1. So if this is the case, we should be able to rescue the phenotype by inhibiting TM1. And we found a TM1 inhibitor, NSC23766. And when we treated cells or islets that were treated with tamoxifen with the inhibitor, we could restore the insulin secretion back to normal levels of the controls that were also treated with NSC 2377, so 23766, which is suggestive of the fact that this is really at the core, the polarity is regulating or blocking insulin secretion via TM1, RAC1 dependent actin reorganization and internalization. So there have been other studies linking epithelial-mesenchymal transition of beta cells with type 2 diabetes, and shown here are just two of them. So we were wondering if there's really a link to type 2 diabetes, not just to our models and insulin secretion, but also there's any evidence in human patients. And we looked into islets that we got from Pat McDonald's in Alberta, from the Alberta Islet Corps. And what we found was when we manipulated IFT88 and depleted them in these islets from donors, we see a decrease in glucose-stimulated insulin secretion in each one of these different preps. Maybe even more important, what we found was that in 3 different preparations of islets from type 2 diabetic donors, we see an increase of phosphorylation of FA3 compared to the controls that we had. In this case, we compared them to 2 different non-diabetic controls. And we see and we see an increase in the phosphorylation. In collaboration with a team from Tübingen, we looked into a very small cohort of partially pancreatectomized patients, and we found that there was a negative correlation for fasting glucose and HbA1c levels and the expression of RAB3IP, also called RABin8, which is a direct interactor of BBS1 and BBsome, and is linking the BBsome to vesicular trafficking. And there's a positive correlation of RAB3IP with HOMA2B, both for the C-peptide and insulin, suggesting that there is really some link. And we're pursuing this in collaboration with Michele Solimena and the IMIDIA consortium, that is a large collection of RNA from laser capture microdissections of partially pancreatectomized patients. What we found was there is a significant decrease in IFT52, which is a direct interaction partner of IFT88, and in ARL6 or BBS3 and BBS2. And BBS3, shown here in pink, is actually interacting with the BBsome, mediating conformational change. BBS2 is in the top lobe here, showing that meaning that there's also involvement of BBS2 in the conformational change upon binding of ARL6. Maybe even more exciting, we see a strong positive correlation of HOMA2B with CIS1 and a negative correlation with HbA1c across all different patients, suggesting really that there might be not just correlation, but also cause in ciliary function. CIS1 has been shown in 2009 to be a bona fide ciliary gene. Another line of investigation we carried out in collaboration with Annette Schürmann's lab and Charlotte Ling in Lund University. And what we did there, we compared the gene expression in diabetes resistant versus diabetes prone mice and compared them to type 2 diabetic islets and non-diabetic control islets from human donors. What we found was when we were sparsing that for ciliary and annotated genes, we found 81 different genes that are co-regulated or similarly regulated in the mice and the humans. And almost all of them were actually indicated with ciliary disassembly and cell cycle progression, suggesting that the cilium is also important for beta cell maintenance and proliferation. So moving on now from the published data to the unpublished data, this has been done by a stellar postdoc, Jan Schong, in Per-Olof Berggren's lab in Stockholm. So this was a collaboration. And Jan started looking into the intra-islet capillary density in BBS4 knockout animals. And what she found both in sections as well as in isolated islets was that there was a reduced intra-islet capillary density in these animals compared to the wild-type controls at an age of 3 months. Parasite coverage is normal, so this doesn't seem to be at the root of the observation. And when she transplanted the islets isolated from BBS4 mice into the anterior chamber of the mouse eye to follow them longitudinally, she could also recapitulate the phenotype. When you compare here the wild-type to wild-type transplantation to the BBS4 to wild-type transplantation, you see that there is a significant delay of vascularization, which catches up, but there seems to be still larger diameter of the intra-islet capillaries compared to the wild-type case. And you can see this here in quantification of the revascularization rate and also the diameter. So to show that this is not only cilia related or not only BBS4 specific, but it's really truly cilia related, we also looked into a model that was given to us by Heiko Lickert's lab, and we found that PFO mice or pitchfork mice also have reduced intra-islet capillary density and increased diameters in the sections of PFO mice. Transplanting islets from PFO mice into the wild-type mice gave a similar phenotype, albeit less severe compared to the BBS4 mice. But importantly, they catch up at some point, and there's no important difference, similar to the BBS4 phenotypes. So to figure out what the problem is, or whether it's more the beta cells and the cilia and beta cells, or the cells, the endothelial cells with their primary cilia that are causing this defect, we capitalized on the fact that after prolonged time in culture, so this is at day 7 of islets in culture, we lose endothelial cells. So at day 2, there's only clusters of these, but really all endothelial cells are lost after prolonged times in culture. So what Jan did was she waited between 2 days and 7 days in culture for the engraftment of these cells. So after short periods of culture, tissue culture of the islets before transplantation, we see a significant contribution of the donor endothelial cells in the vasculature of the engrafted islet. And after 7 days in culture, there's really predominantly recipient endothelial cells found. And when we did this and transplanted BBS4 and wild-type islets into the eye of wild-type mice, after 7 days in culture, we did not observe a significant difference, both in the speed of revascularization or in the diameter of the blood vessels, suggesting that it's really the primary cilia on the endothelial cells that drive the phenotype. So we reversed this experiment and transplanted wild-type islets into BBS4 knockout animals. These are not diabetic by normal standards. So the random fed blood glucose levels are normal in the BBS4 animals. However, they're significantly obese. So they're heavier than the wild-type controls, and they're also glucose intolerant. But the beauty of the approach that we're choosing, or Jan is choosing, is that really mouse have 2 eyes, which means that you can use one eye for the transplantation of wild-type islets, and you can use the other eye for the transplantation of the BBS4 knockout islets, so you have an internal control minimizing the individual differences. And what Jan found that after 2 days in culture and after 7 days in culture of BBS4 recipient and wild-type donors, there's a significant delay and higher diameter in the grafts of the wild-type islets that were cultured for 7 days. So in this case, we have really a large contribution of BBS4 knockout endothelial cells compared to this phenotype, where we see really still a lot of wild-type endothelial cells contributing to the vascular phenotype. And this was quantified by Jan again, and you can see that there is a defect or a delay in revascularization of BBS4 versus wild type, and that this gets worse the longer we keep the islets in culture. Also, the diameters are increased after prolonged culture of wild type islets. By contrast, we also used, of course, our BICKO islets, and what we found was that there's a negligible contribution by beta cell cilia. So this was done by Niels O'Brien staining. He used PCAM1 as a marker for endothelial cells and found no difference in vascularization of the BICKO islets. And also when we transplanted BICKO islets into the anterior chamber of the eye, we did not see a significant difference both in the diameter or in the time that it took to revascularize. Why is it important how fast we revascularized? It has been shown that there is a correlation. So the faster an islet graft or any kind of transplantation graft is revascularized, the better is the graft survival. So in the context of beta cell replacement or islet replacement, it is actually important how well the islets are revascularized. So we looked also into the barrier function or vascular function in general of the intra-islet capillaries. And what we found was that there's an impairment. For example, when we look into the fenestrae of the intra-islet capillaries here in the wild type animal, there is a strong reduction of the capillaries or fenestrated capillaries in BBS4 knockout grafts that's quantified here, but there's actually no difference in the basement membrane thickness. And functional assays, of course, we could carry out because this is a live animal. And what Jan did was inject fluorescently labeled dextran and then looked at how fast it leaked out around in the area around the islet. So in this area here. And what she found was that there is a significant decrease in the kinetics of fluorescent leakage in the BBS4 transplantation case compared to the wild type islets, suggesting that there's really a delay in nutrient delivery. And to confirm this, what she did was use a fluorescent analog of glucose, so we can actually directly measure how fast and how efficient glucose is transported into or across the vascular barrier. And she found that there is a decreased number or a decreased concentration of fluorescently labeled glucose that is leaving the intra-islet capillaries, suggesting that there's a defect in nutrient delivery. And it could also mean that there's a defect in insulin disposal to the peripheral tissue via the bloodstream. When Jan transplanted the critical number of islets into the mouse eye to rescue streptozotocin-induced diabetes, what she found was that after 4 weeks and after 8 weeks, there was intolerance, so the B4 islets were not as effective in rescuing the phenotype. And after 12 weeks, there is a delay. The overall glucose tolerance is normal, but especially in early times at 15 and 30 minutes, seems to be a problem with the disposal of insulin or nutrient delivery, which could be consistent with what she observed before. And we know that islet fenestration or capillary fenestration is linked to VEGF-A signaling and VEGF-R2 signaling. And so we looked in, or Jan looked in endothelial cells and primary human endothelial cells for the that were depleted of BBS4 for the expression of common vasculogenesis regulators such as Ty2 and eNOS, but she only found reduction in VEGF-R2, the receptor for VEGF-A, Ephrin A1 and Ephrin B2. And when she looked into the activation of VEGF-R2 at the tyrosine 1175, she found a blunted phosphorylation, so a blunted activation of VEGF-R2, the receptor for VEGF-A, and this is consistent with a loss of downstream signaling such as PLC, PAKT, or MAP or kinase pathways. when she quantified, I mean, we've shown that VEGFR2 is reduced when you quantify the phosphorylation over the protein levels of VEGFR2, we still see reduction in the signaling pathway. So knowing what we know from Francesco's work, I was really wondering and reading up on the signaling pathway of VEGF-A, VEGFR2 dependent signaling, and it turns out that VEGFR2 needs to be internalized to start signaling. So it does not signal from the plasma membrane, but only from internalized endosomes. So what Julia did in this case was she supplied labeled VEGF-A to a pancreatic endothelial cell line, MS1, and what she found was that if she depleted these cells of BBS4 or IFT88, there was significant reduction in the internalization of these endothelial cells. And this was also found, Jan did this study, she looked into the internalization of VEGFR2 directly by not permeabilizing the cells and only staining for membrane-bound and surface presented VEGFR2, and there was a significant delay in the internalization of VEGFR2. This is consistent then with functional tests that she did. So proliferation, for example, significantly reduced, and the Scratch assay shows a reduced migration distance of the nuclei, and also tube formation assays, both in tube length and branching. Jan found significant reduction when she depleted endothelial cells of BBS4, suggesting that this is really, again, endosomal processing that is disturbed by the absence of IFT88, which was what Julia looked into, and BBS4. So the last couple of slides, I just want to briefly give an outlook of what we're going to do in the lab as soon as we open up again. And this is primarily Julia's project. So Julia, as I said, has done the pulse chase surface biotinylation assay and found this defect in recycling endocytosis. And one of the hallmarks or gold standards of recycling endocytosis is, of course, transferrin and transferrin receptor. So she looked in different cell lines and different tissues and found reproducibly a reduction in the rate of uptake and the number of compartments that are occupied by fluorescently labeled transferrin when she knocked out IFT88 and IMCD3 cells in MS1 cells, and this has actually been done by Francesco still in Min6 cells. So if this is the case, what would be the physiological phenotype that is associated with it? I was thinking into it and going back a little bit to my genetics training. So I was looking up the online Mendelian inheritance in men, phenotypes for mutations, for example, in the transferrin receptor. And what turns out is that this is associated with hemochromatosis. It's a defect in iron metabolism. And so we looked into the blood chemistry of BBS4 knockout animals, and what we found consistently was a reduction in the unsaturated iron binding capacity and the total iron binding capacity, and a small but not very dramatic difference in transferrin, suggesting that this is really more global, not just referring to FA3 and ephrin signaling, but also to transferrin receptor. In collaboration with Francesca Sacco and Matthias Mann's lab, we've also done phosphoproteomics in BICKO islets, and you've heard about this from Natalie Kramer last Friday. Francesca found 10,206 phosphosites in these islets. Out of those, 2,590 were significantly misregulated. In absence of IFT88, only 143 of those were increased, and those were associated with exocytosis, snare, and syntaxin, but really the predominant phenotype was a decrease in phosphosites, and almost all of those were involved in phospholipid binding. So this is really something that we want to understand better, and we have a couple of ideas that we're looking into. So it's really, we're itching to get back into the lab, as I'm sure most of the audience is. And finally, our latest addition to the Gerdes lab is Priya, and Priya is capitalizing on the internalization as functional readout. So she has developed a high throughput screen assay based on transferrin uptake. And a secondary assay will then look into the uptake of different other ligands. And she was ready to screen and test the assay with a library of FDA-approved drugs, which was in the week before the shutdown. So now we at least know the conditions for this assay and hopefully can pursue the screen once everything is back to normal and this is over. And with this, I just want to briefly summarize the findings. So first, the targeted disruption of beta cell cilia is important for glucose tolerance. If they're disrupted, they lead to glucose intolerance. And also beta cell maintenance is impaired when beta cell cilia are lost or dysfunctional. We've shown that hyperphosphorylation of FA receptors is blocking insulin secretion in the islets of tamoxifen-treated BICKO animals. Cilia and basal body dysfunction is involved in endosomal processing. I've shown you two different examples, both with FA and VEGF-A. Also, cilia disruption triggers EMT-like polarity defects, and there's now some additional data from other groups showing that cilia are really involved in maintaining polarity across the EMT spectrum. Jan has shown that endothelial cilia modulate the intra-islet vessel density, and I've told you why this might be important. The depletion of cilia and basal body components blocks VEGF-A and VEGFR2 signaling, at least partially because VEGFR2 does not signal from the plasma surface or the cell surface, but has to be internalized in an F1B1 dependent process. There's human evidence that cilia and centrosomal genes are linked to beta cell proliferation, and that IFT88 depletion in human islets also recapitulates the BICKO phenotype. And last but not least, CIS1 transcription correlates with diabetes progression in the islets of partially pancreatectomized patients, which could suggest that there is not only correlation, but also causation. And with this, I want to thank again people in my lab who've been doing the bulk of the work, and our collaborators, both at the Helmholtz and nationally, as well as internationally, namely Jan and Pio Berggren, who has really done a super job in trying to figure out the role of primary cilia and endothelial cells of intra-islet capillaries. And with this, I want to thank you for your attention. And I'm happy to take questions on Twitter. And when all this is over, I hope to see you in real life and maybe discuss some projects. Thank you.